So a very warm welcome to the COG-UK first anniversary science showcase today. So my name is Sharon Peacock. I'm the director of the COG-UK Consortium, and I really do want to offer you a very warm welcome. My welcome also extends to international attendees today with registrations from 41 different countries. So as reflected in the title of the meeting, COG-UK is 1 year old this week, and our science showcase is an important part of our reflections on the past 12 months. So turning then to the meeting, given the time constraints we have today, we've considered our programme very carefully, and given the prime importance of variants, we have constructed our meeting around these. So we have 3 sessions. The first is on new insights into molecular tests and massive sequencing. That will be chaired by Dr. Catherine Ludden from the University of Cambridge. The second session is on the distribution, detection and prioritisation of SARS-CoV-2 variants, chaired by Professor Emma Thompson from the University of Glasgow. And the third session is on the variants in relation to patients and prevention, and that will be chaired by Dr. Sally Corden from Public Health Wales. So there'll be 5-minute breaks between each of the sessions to allow you to stretch your legs if time allows. And there will be questions in between the talks, again, if time allows. So I'll finish with a few housekeeping notices before I close. So first of all, the event is being recorded today. Secondly, as we go through the afternoon, I would encourage you to put your questions in the Zoom Q&A box. And I've said that there may well be time for questions, but we would like to hear from you as the talks progress. So I hope you enjoy the meeting and let's begin. I'll hand over now to Dr. Catherine Ludden to begin session 1. So thanks very much and do enjoy.
Thank you very much, Sharon. It's a pleasure to be chairing the first session, as Sharon introduced, which is on SARS-CoV-2, new insights into molecular tests and massive sequencing. And our first speaker is Dr. Cordelia Langford from the Wellcome Sanger Institute, who, as we know, has been a major player in massive sequencing, and I'm sure has a lot to tell in this area. So Cordelia, over to you, please. And for the questions, can I, as Sharon mentioned, can you please put them in the chat? We will address them as we're going through, and if we've got time after each session, we'll go through any questions then. Thank you.
Thank you very much. I'm just going to share my screen to show my slides. And I'd like to start by thanking very much the Sharon and the organisers for inviting me to speak today, and I wish all our colleagues in COG-UK a happy anniversary. On March the 23rd last year, Sanger began our preparations to sequence the genomes of SARS-CoV-2 to feed into the genomic surveillance goals of COG-UK. This activity, which is termed Project Heron internally, is a project of inspiring and ambitious vision. And we believe it very much plays to the strengths of our organisation, and it's being achieved by pivoting our technology and our expertise within both research programmes and our operational pipelines to rapidly deploy an effective and functional response. This was a huge logistical, intellectual and technical operation, which was set up from scratch pretty much in a matter of days and weeks. It was initiated by establishing a vision first, and we established also the framework for a plan, which was designed and implemented through a highly collaborative effort through a multidisciplinary fully integrated team, a team who shared resources and used agile methods to progress towards our goal. Our overarching strategy was to repurpose our existing pipelines, certainly in the first instance, to stand up essentially a minimal viable product as soon as possible, and to also in parallel develop a roadmap to solve the issues that we faced and to refine and scale that effort iteratively. We established a formal plan quite quickly with work streams established and with milestones and clear strategies in order to capture and define our goals in the short and also in the longer term. Initially, our goal was to process primary swabs from pillar 1 testing, and having established some capacity, this was a brand new activity to achieve this primary swab processing. We actually evolved and developed our strategy and subsequently transitioned towards processing RNA residues, so pre-extracted. And these were actually waste products from the pillar 2 lighthouse testing. And the process involved the consolidation of just positive samples. Having stood up sufficient capability and processed our very first 100 samples, which are pictured here in the bottom left-hand corner, we submitted that very first data to CLIMB, and we reflected and started immediately to seek to improve our offering. We then designed, tested, and implemented iterative enhancements to realise incremental increases in our capacity and throughput, as reflected in this graphic shown here, that show monthly totals of samples that were processed through the Project Heron pipeline. By planning and resourcing our technology developments, we were able to realise these iterative increases. We've also, over that period of time shown in the graphic, reduced the turnaround time taken to generate and upload data. And we've also sought to improve the quality of the product generated. And our efficiency was very much improved through a number of efforts, but in particular by streamlining the sample supply. And to do that, we forged and built really strong partnership relations with external stakeholders. These are groups and organisations we hadn't necessarily worked with before. We established a partnership approach to really break down barriers and to progress together. We maintained motivation in staff by jointly marking each of those milestones, and we became very good at recognising and celebrating our successes, whilst at the same time recognising everyone as an individual for the contribution they've made. Last week, we actually reached the milestone of sequencing all of the positive samples that were supplied with a CT value of less than 30. Also, our 20 millionth sample was received, and we also passed a milestone of having submitted more than 200,000 genomes, sequenced as part, of course, of the wider COG-UK success and effort. And the Project Heron team themselves are now processing 20,000 samples a week, and we've developed a roadmap to scale further to first to 25,000 and then subsequently up to 50,000 per week over the coming months. To provide a robust and sustainable platform, we established a national logistics system and infrastructure with full end-to-end operations and with an analysis capability. This starts with the sample shipments across each corner of the UK, and also includes the timely access to relevant plate data and metadata. The ability to place staff into the Lighthouse labs, and also moving to daily shipments smoothed out what had previously been quite a lumpy sample supply. Access to the plate and to the crate manifests, as well as to individual plate maps before shipments arrived, significantly streamlined our processing times and actually cut our waiting time down. We initially relied on scientific operations staff within the Sanger Institute, as well as researchers and a large number of volunteers actually from across Sanger. But we've now built a fully dedicated capability, which is funded by NHS Test and Trace, with more than 40 dedicated staff, completely dedicated lab equipment, refurbished, custom-built laboratories. And those teams are working 7 days a week now. The national logistic infrastructure is underpinned very much by the need for digital connectivity and also very strong and robust external relations. So the digital integration itself required multiple complex data feeds coming both from private organisations as well as government organisations. And the sample logistics element required the build of a new software tool that enabled the full end-to-end tracking of samples as they flow through the process, but also had to integrate across our multiple LIMS systems and warehouses that are already in place at Sanger. Throughout this project, in addition, we've embedded a new culture, and that is of lean-based process improvement, which has enabled us to dramatically reduce our turnaround times and elements of waste through that end-to-end process. Key enablers for us have been those of our supplier partnerships and relations, as well as academic collaborations and innovation, both from our partners and from our internal staff. We've had the benefit of having access to loaner equipment, which have enabled us to scale rapidly. We had the benefit of collaborating to code new robotics programs to establish and streamline our processes, and also have had the provision of insight and reagents of our commercial partners to accelerate our protocol development and the implementation of those. Throughout this activity, we've driven a new culture of empowering and recognising our technical staff, and that has occurred at all levels in the organisation. I mentioned earlier that more than 300 people at Sanger have contributed in some way to Project Heron, and their skills, knowledge, dedication and hard work has been highly valued and continues to be so. The work of our technicians and staff has been spotlighted through various celebrations and media events, and we're so proud of our staff and what we've collectively achieved. Together, I think I could summarise it by saying that we've collectively translated an incredibly ambitious vision into everyday reality. Thank you very much for your attention, and I will stop there.
Thank you very much, Cordelia. That was a wonderful session. It really highlights the immense effort by such a huge team at Sanger. I think trying to capture in 10 minutes the work that your team have done, from testing and working in the Lighthouse lab to sending the samples, the data, processing, sequencing and analysis. I think it's such a huge challenge that you've taken upon yourselves, and definitely something you should be very proud of. And I can hear that proudness in your voice as well. So just a massive achievement. I have one question here from the chat, so I'm going to pose it to yourself, and I may follow with some questions if we've got some time. The first question is, what is the estimation turnaround time at the moment from swabbing the patient to sequencing and analysing the genome and identifying mutations such as VOX and VUIs?
That's such a good question, and of course it's a question with many different answers depending on which element of the process one looks at. I mentioned earlier that we're very much considering the full end-to-end process, which would include, actually, from our perspective, the point where a sample has actually been tested as positive. So unfortunately, I don't have insight into the process for taking a swab and actually performing the test itself. But in terms of the end-to-end process from a sample, an RNA sample having been tested as positive, the turnaround times are sort of very much started out really quite long to begin with. So about a year ago, we were looking at maybe 15 or in some cases 20 days to get that full provision of not just data, but having variants identified and analysed, as well as the relevant metadata itself. And in fact, the turnaround times have technically have reduced quite significantly, down to about a quarter of that time now. But we do still have bottlenecks where there might be constraints in the process that might be due, in some cases. For example, we had to slow things down over Christmas because of a global supply chain issue that meant that we couldn't process anything like at the speed that we wished to. But also, I'd like to reference back to that digital connectivity, which is absolutely key to enabling the data itself that might be generated in any sort of quick time frame, but it's only relevant and meaningful for interpretation as long as it's got the relevant metadata associated with it. So there are ongoing challenges of ensuring that the data is usable within a really effective and meaningful time frame. It's a great question, thank you.
Thank you, Cordelia, and I think you've really well covered that there's so many different components to actually the turnaround time that it's not as straightforward as some people may expect, and also there's various people working on different sides of it. So thank you very much. I have another question from the audience. It is, can you talk about how surge testing / sequencing is managed compared to standard pillar 2 samples?
Yes, I'd be very happy to talk a little bit about that. There is actually, there's been a number of iterative approaches to support the effort of sequencing the surge testing. And one of the key enablers was to implement a system where samples that were surge tested were actually consolidated at the Lighthouse labs before they were sent to Sanger. This cut down, or cut out, if you like, quite a big chunk of time that enabled a much more agile response and the ability to generate that data more quickly. So the process itself, at a reasonably high level, would involve the cherry picking or the consolidation of those surge test positive samples into plates that are then sent through the daily shipments to Sanger. And then there's a slightly different pipeline that the samples are processed through at the front end of the pipeline, because instead of going through and feeding into our really large scale cherry picking front end, we actually send those surge plates through to a process which is less complex, which we call stamping. What that actually means is that we use liquid handling robots to take a very small aliquot of each sample and make a copy of that plate, which is then fed into the downstream sequencing process.
Thank you very much, Cordelia. I have one final question, I guess, to summarize everything that you've achieved and the difficulties is, what would you describe as your biggest challenge that you've had to face and overcome to deliver this massive sequencing?
That's the sort of question that I usually would ponder and request to reflect on overnight. But one of the things I'm going to say is that I think perhaps from a personal perspective, what was quite unexpected was the sheer scale of the sort of number of organisations across the UK that in the first instance it would have seemed an intimidating challenge to enable and find ways to work with them, to coordinate, liaise, build these brand new partnerships. But actually, here we are a year later, and actually have achieved together collectively what might have seemed like a little bit of an impossibility in the start. And if people wanted to sort of say so how was that achieved, I think all of us probably would reflect on the fact that we've sort of broken challenges down into their component parts, and also collectively and inclusively workshopped, brainstormed, tried to find solutions together, and work, yes, essentially work them out together.
Great, thank you so much, Cordelia, and definitely on the spot question there. And just for me, on behalf of COG-UK, just wanted to say a massive thank you to yourself and all your team at the Sanger Institute. Your contribution has been immense, and will be for the year, and many tests and sequences to come. So thank you very much.
Thank you very much, and it's been a privilege to represent the Project Heron team. So thank you again.
Thank you, Cordelia. And I'm sure there'll be many questions in the chat box as well to follow. But in the interest of time, I'm going to move over to our next speaker, which is Dr. Kate Templeton from the University of Edinburgh, sorry. And Kate is going to speak to us about SARS-CoV-2 mutations and diagnostic test accuracy. And just as a brief introduction to Kate, she's one of the PIs for COG-UK, and is also a consultant clinical scientist in the Department of Medical Microbiology at the University of Edinburgh. So Kate, we're very looking forward to hearing your talk. So over to you. Thank you. And just to know, Kate, you're on mute.
So lots of Kates and Catherines at all times. Thanks so much. This is quite a short talk, just talking about how different mutations have effects on our PCR, and how we've used COG, the data generated by COG, to improve this work. So oh, my slides are a bit slow. So this is what occurred in September, October time. We started to see a phenomenon with particularly with the TACPath assay, which is used in most of the UK government sites, where you would have 2 genes positive and the S gene was negative, and this linked to the presence of, also linked to a clinical scenario of the new VOC. So the S gene being negative also showed that while the diagnostic test didn't work, it also linked to the appearance of this new variant of concern. And this is just looking over time in Scotland what's happened. So here's all our different health boards, and with the different S gene over time. So this is what we started to see in October, November time. So around about between 30 to, or 20, 30, 40, 50% at that time had this variant of concern. And now this is looking in end of March, and we're now up at 80%, even in some cases, Highlands up to 100%. And so really there's a lot of places, and the overall for Scotland is around about 90%. So this is us being able to use an assay to monitor a variant of concern, but ultimately the whole thing was brought out by the fact that the assay had stopped working for one gene. And because the assay had 2 other genes, it wasn't too much of a diagnostic problem, but if it potentially was to lose more of those mutations, the assay would stop working. So what we then said, I said to all the registrars that are signing out reports, I'll give you a Mars bar for every time that you see one of these variants popping up, and then we will get it sequenced. And lo and behold, the next day I get told, here's this one, Kate, do you think we should highlight and get this one sequenced? And so here, just to run through this, this is a report of how it looks for us in the laboratory. So here we can see that we've got ORF1 that's coming up clearly very strongly positive. The N is completely negative and the S is positive. So not the same phenotype at all as the S gene dropout. And what we did was we sequenced that, and it was the B1177 that had been around since October, but it had 2 mutations in the N gene that were 9 base pairs apart. And that would be enough, if that's where the probe was binding or the primers were binding, to affect that assay. And so what we then did was we then looked through all the data that we had on the TACPath assay. So we had a lot of positives, and we found that 0.1% of those had, so 120 had this phenotype. We looked across all the different health boards and could see that some, so Tayside, which is where we first picked it up from, had maybe more than, say, us in Lothian having slightly less. And then what we did was we looked at this over time. So it's started to emerge towards the end of December, and and then by January, February had largely disappeared, and that was probably because the dominant phenotype by that time was the VOC1, so we weren't seeing this one as much. And then we'll ask the question, are there other assays that need to worry about? And this was published in Journal of Clinical Microbiology by sort of an assay CEFID. And again, here they had an N weak positive and an E strong positive. And again, they were able to show that there was some mutations associated with that. And this is just looking at a snapshot of all the different commercial assays that we use across Scotland. And so you can see there's a lot of different manufacturers, and they all target different genes, and they all have different primers and probes. But we in the because the companies are the only ones that know the primers and probe sequences, the only ones we know the primer and probe sequences for are those that are in-house assays. So we ultimately just need to have a mechanism that we can keep monitoring to see if we're seeing a difference between the different sequences we're getting for, say with the Altona, if the E was strongly positive and the S was negative, that we then prompt that into the sequencing and then have a dialogue with the companies. And we then need to have a sort of real-time rota for looking at all these different sequences, and not just probably putting it down to an individual noticing it and my Mars bar approach. So we've now been
So Kate has frozen on my screen. So Kate, just to let you know, we can't hear you at the moment. So we'll just give you another minute or two, and if necessary, you may need to log out and log back in again. So in the interest of time, I'm going to move to our next speaker. As I said, please post your questions for Kate in the chat box. We will pose those questions to Kate and then come back to her at the end to see if there's anything in addition that she would like to add to her talk. So in the interest of time, I'm going to move over to Dr. Leigh Jackson, who works as a scientific project manager with COG-UK and also a lecturer at the University of Exeter. And Dr. Leigh Jackson is going to speak to us on non-sequence based molecular methods to detect SARS-CoV-2 variants. So over to you, Leigh. We're very excited to hear your talk and to learn about the new methods coming through. So thank you very much.
Thanks, Catherine, and thank you for the invitation to speak here today. Just want to check that everything's okay with my slides before I carry on.
Yes, perfect.
Yeah,
we can hear you and see your slides, so thank you.
Fantastic. Okay, so I'm going to talk today about some non-sequence based methods for detecting variants, and it's a bit different from all the other talks you'll be hearing today. So first, to start off with, what is the importance of detecting variants? And I think most people will be aware of this, so I won't spend too much time. But here I'm showing our latest coverage report. So I noticed in the chat someone asked about kind of surveillance and coverage. And what this map shows is the percentage of samples that test positive that we have sequence data for this week. So as you can see, for the vast majority of the country, we have in excess of 20% of all positives, and in a lot of cases it's far more than that, and we're now asking all samples to be sent for sequencing. So we're really increasing our coverage here. That allows us to have an overview of the circulating lineages, to track different emergence of mutations evolving, and then the kind of thinking behind that, the importance behind that, is to identify these individuals with variants of concern, link back in a timely fashion to the public health agencies, so that they can follow up and stop these potential outbreaks. So one potential method that's being trialled at the moment is the use of variant genotype assays. So rather than the full sequence being generated, this would be an add-on to the diagnostic test, which would aim to pick out some specific variants or mutations of concern. So in the last couple of months, a number of commercial providers have started developing assays aimed at detecting these specific variants, either individually or in combination. And the way these assays work is by using allele-specific primers. So there'll be a mixture of primers in the reaction, one that would match up with the variant or the mutation that you're interested in, and the other for the, let's say, the normal, the wild type sequence. And that will give you a quick read of a select number of mutations, and might allow you to assign lineage based on those combinations. And as you can see on the right hand side there, this is done by different signal intensities from the different coloured probes, and we'll come back to that a bit later. So what are the possible advantages of doing genotyping over sequencing? So potentially there may be a quicker turnaround time. As you've already heard from the Sanger workflow, the samples have to flow from a diagnostic lab to a sequencing centre, and then go through the pipelines there, so that can take a few days. So this could allow a much quicker turnaround time for getting some kind of indication on mutation or variant status. Because it could be added on in a kind of PCR-based way to the existing test, this could be scalable and done at high throughput relatively quickly. There is a potential for working with the different companies to design new assays as and when new variants pop up and new mutations pop up. And the individual cost per sample is obviously cheaper than sequencing the whole genome. So I said possible cost savings, and again, we'll come back to that because it's a little bit more complex. So thinking about the other side of the coin, so the possible disadvantages of genotyping, it might not seem obvious, but we're of course restricted only to known mutations. So you will lose that surveillance capability if you were to switch entirely to genotyping. And I've shown a screenshot here from the COG Mutation Explorer, just focusing on the 501Y mutation. And you can see that depending on when you designed your genotyping assay, you may or may not have included that location. So going forward, it's always going to be important to keep an eye on what's coming up and be aware that these genotyping assays will only be a snapshot in time. There is also the potential impact on sequencing itself, specifically if we're talking about repeating samples that may have failed sequencing, because adding in an additional test, or in some cases a number of additional tests for the different mutations covered, will have an effect on the amount of material available to be sent to the sequencing site. So that is something we need to bear in mind, and that, you know, inevitably there will be a trade-off one way or the other. So that's something to bear in mind. And also related to that, individual mutations and targets are rarely lineage defining in and of themselves. And so you might need a bespoke combination of those to give you all the information you need. And then, as Kate was talking about, if some of those drop off or don't work, then the interpretation becomes slightly more difficult again. So I just want to talk about one specific issue in utilising this type of technology. And this fits in with work that I've recently published with my colleagues at the University of Exeter, in that there are some inherent issues with using genotyping assays to call extremely rare variants. And the work we did was in human samples, but the kind of the theory translates across pretty closely. And this can be the case even when the tests themselves are extremely accurate. So this is not a problem of the test accuracy. It's as much of a function of the rarity of the variants themselves. So hopefully you can see from the graph that the way that the genotypes are called by these types of assays are by clustering together the different signal intensities that we saw in the first slide. And when you've got a lot of alternate alleles, then so the mutations that you're interested in, they will cluster together quite tightly, and it's quite easy to see that. If you're down to extremely rare variation, then you might not have a cluster at all. You might have one sample, and obviously that's a bit more difficult to differentiate from a technical artifact. So that's the first issue to be aware of. The second one is kind of related to what's known as the base rate fallacy. So if we take the theoretical example where we have an extremely accurate test that is 99.9% specific for a particular mutation, then it follows that 1 in 1,000 of those tests will return a false positive, which is a very good rate. But if the variation that we're looking for is rarer than that, say 1 in 10,000 tests, then you get to the situation where you have more false positives than true positives. And so then we have to kind of rely on the positive predictive value of these tests. So that's considering, given that you've got a positive result, what is the chance that positive is true? And that might be a better measure when validating some of these genotyping tests to make sure that there is still a cost effectiveness argument and a scientific argument, because obviously the cost per sample being cheaper might not add up if you end up having to deploy a lot of resources for positives that turn out not to be so. So this is where the validation of these pilots will be extremely important to make sure that we're using the correct assays in the correct contexts. And the sequencing will need to support that at the same time to validate as well as picking up on new mutations. And I should mention as well that the nature of this technical issue means that the positive predictive value will change over time for different variants depending on their prevalence in the population. So a genotyping assay for the Kent variant, say, will perform extremely well, whereas for some of the rarer ones, it might perform less well. There are things that the providers can do to get around this: extra probes, extra primers, positive controls. So it's just something that we need to be aware of. So just to conclude, there is clearly a compelling public health imperative to quickly identify individuals with specific variants and take further downstream action. Genotyping assays could achieve this at a low cost and at scale. However, the rarity of the variants themselves may present problems even if the tests are extremely accurate. And sequencing will continue to be required both for the validation of these genotyping assays, but also for surveillance of novel variants, which may or may not then be translated into further assays in the future. So I think I will stop there. Thank you very much. I'll take any questions.
Thank you very much, Leigh. That was a wonderful talk that shows us the other side of the coin is, can we detect variants and lineages from diagnostic testing? And I think you've explained quite well that there's definitely some pros and cons that need to be considered. I have one question from the audience that's just came through, Leigh, which is now non-sequence based variant analysis would definitely provide faster information about prevalence of specific mutations rather than lineage, because convergent evolution of mutations. Is there any data on prevalence of mutations, C frequencies of delta 69 to 70 versus B1.1.7?
Okay, and excellent question. Thank you very much. So the short answer, it's an answer in two parts. Firstly, unfortunately, I do not keep the allele frequencies on the top of my head, so I would have to go and have a look at that. But the underlying answer is yes, that information does exist. So we do have mutation allele frequencies for all of the different mutations coming up. So that can be incorporated into the validation and into the calculation of positive predictive value. And I showed a screenshot from the COG Mutational Explorer earlier on, and you can get all of that mutation frequency information from there, and that's a publicly available resource. So if you have a mutation of interest that you want to look up, you can use that to get the frequency information. So yes, that information is available, and the difficulty comes because, as I said, the individual mutations are rarely lineages defining, as the questioner correctly pointed out. So you have the prevalence of the individual mutation to consider and the prevalence of the variant of concern to consider in combination, and that's where the maths gets a bit more complex when you have to take both of those into account. So you have the potential for calling the individual mutation right and then assigning it to the correct lineage is another challenge on top of that.
Great, thank you, Leigh. I know we've referred to the COG-ME, which is the Mutation Explorer developed by the COG-UK Consortium. So that's a link to that is available on their website, but I'll also check to see if that can be dropped into the chat box so people can look at that link as well that you're referring to, which is a really nice tool for looking up the mutations. We have a second question here that's just come through, Leigh, which is this genotyping assay the same as the approach recently announced by the government?
Yes, it is, in a word. So it's been trialled and rolled out in a number of the Lighthouse labs, and obviously part of that pilot study will include a thorough validation, so we can understand the accuracy and utility of using these. But yes, that was the initiative announced earlier on in the week.
Great, thank you, Leigh. And we've got two more questions, and then I'm going to have to stop because we're going to go back to Kate for a few final bits of information from Kate. So the last two questions, Leigh, is where do you think this assay is most applicable within the current framework?
That's a very good question. So taking into account the inherent issues with low prevalence, and I'm very much going to step outside and say this is my personal opinion and not necessarily representative of any official view, but I would say from purely statistical point of view, you would get best use out of this in a targeted manner. So if you had a population that you assumed to be at a higher risk of having a certain variant or variants, then the test would perform better in those situations rather than a kind of a global screening program, if you will. So I think that's where the most utility will be achieved, is by targeting them at risk groups. And then you will minimize the number of false positives to true positive ratio and hopefully get your kind of your sweet spot of resource allocation versus time saved.
Thank you, Leigh. And on the point of resource allocation, the last question is, what is the price per genotyping assay?
Varies significantly between provider, I would say. I don't think I'm best placed to comment on that. Kate might have a better view for that from the lab's end, but there are a number of different commercial providers that each will have a different price for their assays. Yeah, I'm sorry, I'm not sure I can answer that one accurately. Great,
thank you very much, Leigh. You've definitely given us some thoughts to think about today and question the different methods we have available. Unfortunately, I'm going to have to stop there, but there will be questions in the chat box, Leigh. Okay. So it'd be great if you could continue answering those. Catherine. And we'll move back over to Kate, who is just two more slides to show us. And we've also got a couple of questions for you, Kate. Thank you.
So hopefully my internet will survive. So this is just, I think, where I finished off from previously is this is just showing all the different manufacturers that we have in Scotland, and this is showing the number of different targets they have. So you see how with all these different manufacturers we have a whole range of different targets that we need to keep monitoring. Oops. Oh God. And so, but we don't know exactly where the primers and probes are because commercial companies don't let us know this. The only assays that we do know where they are our in-house assays where we've designed the primers and probes ourselves. So all we can do with all the commercial companies is to monitor the different targets and see if their CT values are consistent. And if one is then negative and the other two are strongly positive, or the other one is strongly positive, then we need to sequence. And it obviously needs to be a combined affair using resources of COG and diagnostic labs linking up together. So the next thing I just wanted to mention was about terminology. And I think it's quite good to try and distinguish between what is a variant of concern, which is a virus, when the virus starts to have a public health impact, as in it's more transmissible or it's having an effect on vaccine, and then these ones that are largely a diagnostic issue of concern. It isn't the fact that the virus is any different, it's just affecting our diagnostic assays. And sometimes with the S gene dropout, you can have a diagnostic issue and a variant, but sometimes they are just diagnostic issues of concern. And I think that phraseology may be quite helpful to people outside the diagnostic world to understand the difference between what we're trying to investigate for the large amount of time here, which is just primers and probes not working and enabling us to try and get our assay back up and working, as opposed to there actually being a clinical imperative around it. So the assurance of assays going forward, we need to keep a strong dialogue with the companies, and there's been a group set up led by DHSC on a VOC assurance group, and so with that there's a work to maintain assurance around these different tests, and then with that linking to diagnostic networks. But as I said, we do have the ability with in-house assays. What we could quickly do is put our in-house assays into this program developed by Glasgow, the COG-UK site, and then instantly show that none of our in-house primers and probes had any problems with any mismatches. And the commercial companies can obviously access this site as well, but that information isn't always squared away for us to be able to understand how the primers and probes are changing and with the way that the virus is changing. And that's where I'll finish.
Great, thank you very much, Kate. And I'm very glad that we could get you back to finish your talk, because there's some really excellent points there that we really need to take away. I'm going to ask 2 questions, Kate, and I know we're running short on time, so I'm going to keep it to just these 2. The first one being, do you have frequencies of failure for various commercial assays in wet lab testing?
Frequencies. So with the different assays, when we start to notice it, then we start to so if we pick up the S gene dropout, so we're looking at the frequency and monitoring that over time. Different assays, I mean, if you look at the C gene one, we very rarely see a failure there because it's got 3 different or 4 different targets. I suppose potentially it's more, we're more likely to see problems with the S gene because it's got more variability, but I hope that's answered the question.
Thank you, Kate. And the second question is, how long did it take to develop your in-house assay primers and probes? Is using in-house kits potentially a viable way to study new variants on a large scale in the future? So 2 questions sneakily stuck into one there. So over to you, Kate.
I have to say that we really didn't design the primers and probes. We just took them from publications and then have put them through all the different software packages to see that we like them. So it doesn't take very long, but the thing about in-house assays is when you're running, as we are, many thousands of tests a day, we really want the automation and the assurance that comes from working with commercial companies. So, and we don't want things to be manually and in-house. And so we want a machine that basically takes everything from start to finish all the way through for a PCR step. So, so while in-house is great if you're doing small numbers, when you start scaling up to thousands, it's not great. And then could you use that to design variant assays? Yeah, there's no problem in doing that. And lots of people have designed in-house assays to pick up the variants. But again, it will come down to a numbers thing. If you're having to test many hundreds of samples, and it's the assurance and ISO 15189 accreditation and all of that that pushes us down the route of using the greater quality that comes from commercial companies as opposed to doing it with the in-house.
Thank you, Kate. I'm sure there's many questions that will come through the chat box, but in the interest of time, we have to move to the next session. There will be a 2-minute break between now and the next session to allow you to stretch your legs. And the next session will be chaired by Professor David Robinson, who will be focusing on the surveillance of the variants. So thank you very much, David, for agreeing to chair the next session. So we'll commence the next session in approximately 2 minutes. Thank you.
Good afternoon. Unfortunately, Emma Thompson couldn't make it to chair this session. So my name is David Robertson. I'm also based at the CVR in Glasgow. And so then the next session, we're going to look at some of the variants and their distribution and think about how the variants and mutations are prioritized for tracking. And so the first speaker is Áine O'Toole from the University of Edinburgh, and she's going to give us an overview of the global epidemiology of SARS-CoV-2 with the focus on the lineages of concern.
Unmute myself now and share my screen. Okay, I hope you can all see that all right now. I wanted to first thank the organisers for the invite to speak at COG-UK's 1-year anniversary showcase. It feels a lot more than 1 year has gone by since all of this work began. I wanted to start with acknowledgements to emphasise how many people and teams were involved in this effort to track new variants of concern over the last few months. And of course, COG-UK is here at the top, but it's really been a concerted global effort with scientists across the world generating and sharing sequence data as soon as it's been produced. We can develop tools to track variants of concern, which is what I'll be telling you about today, but we continue to rely on the cooperation and openness of scientists and public health agencies around the world. So particularly in recent months, we've become more and more concerned with identifying new variants or lineages of concern, and then tracking their spread both nationally and around the world. And to help do this, and at the time we were focused particularly on B.1.1.7 and B.1351. On Christmas Eve, Verity Hill and I wrote a tool called Grinch, and we called it Grinch because it was built to track the lineages that had stolen Christmas, as was in the papers at the time. And essentially how it works is we download all GISAID SARS-CoV-2 submissions every day. At the moment, we're now at 871,000 SARS-CoV-2 genomes submitted to GISAID, which is an incredible number. And we run pangolin on them to assign the most likely lineage to all sequences. And just to give a bit of background, pangolin is a tool we wrote about 10 months ago now, and that's been used across the world to assign lineages to SARS-CoV-2 genomes. Lineages are ways of grouping the diversity of SARS-CoV-2 into phylogenetically linked epidemiological clusters. So for instance, these particular lineages of concern we're talking about today. So pangolin can help researchers assign lineages to newly sequenced genomes. And we've actually recently reached our own milestone. Just this week, we've had over 1 million sequences now queried using the Pangolin web application, which is insane. So in terms of automated pipelines, Grinch is quite simple. It runs the download, the quality control, alignment, and runs Pangolin, and then gathers all this information back together and produces a report that we share publicly every 24 hours. And we've been hosting these reports on the COV Lineages website since the beginning of the year. And if I just use B117 as an example of the sort of information that we have available on this website. Now, I'm sure absolutely nobody has escaped hearing about B117 over the last few months, but just to recap, we've got a lineage that arose in September last year, characterized by this burst of mutations at the base of the lineage, some of which are mutations that are very likely to have biological significance. And there's evidence of this lineage having increased transmissibility, and we're seeing this continued now. We've been tracking this lineage since December, and as of this morning, there are over 200,000 B117 sequences on GISAID from 102 different countries. And this map shows, it's colored by date of earliest sequence per country, the darkest countries being the earliest. And because this is a showcase reflecting on what's happened this year, I went back to the report on Christmas Day, when B117 was in just 8 countries around the world, and all bar the UK had only just detected imported cases rather than local transmission. And then today, the picture looks very different. The vast majority of B117 sequences on GISAID are still from the UK, because the UK does so much sequencing partly, and counts of sequences that we see are very much a product of the genomic surveillance of that country. So there's still quite a few countries now, though, that are reporting local transmission of B117, and that continues to rise. If we look at the frequency of B117 now, this is a little bit messy now because there's so many countries with it. We're calculating the frequency by saying what proportion of sequences from a country are B117 since the first B117 sequence was detected in that country. And for most countries, it's really quite difficult to say anything meaningful about the frequencies. So if I zoom in to just look at the highest frequencies here, for a lot of these countries, the overall sequence counts are very low, often only a handful of sequences. So many countries are targeting cases that are specifically associated with travel, particularly travel history from the UK. So that doesn't necessarily mean that that country has 100% frequency in the community. And that's just one of a few biases that we're aware of that are associated with the data that we see representing SARS-CoV-2 on GISAID. So Like I mentioned, there's genomic surveillance intensity differences. Countries may be targeting travelers, countries may be specifically targeting this spike gene target failure. And also there could be a bias in uploading to GISAID. So potentially variants of concern may be more likely to be uploaded because they're potentially more interesting sequences or something like that. We also know that the UK has approximately a 2-week lag in community sequencing and getting up onto GISAID. So this may be true for other sequences, and we do see this lag in sort of recent data doesn't necessarily indicate that the cases of B117 are dropping. So we have this as well. We're aware of these biases. So to account for some of them, we've got our hands on flight data, and we have actual passenger volumes from London airports from back in October, which gives us an idea of the actual movement of people and may help account for the differences in genomic surveillance efforts in different countries. So when we got this flight data, this was what it looked like back on the 27th of December. Here we're showing any country with more than 5,000 passengers leaving London in October. You can see the top 3 destinations are Spain, Italy, and Greece. And at the time, we didn't notice a huge correlation between number of sequences on GISAID, which was quite low at the time for B117 in general, outside of the UK, and number of people. But since then, slowly but surely, we've seen these countries report cases of B117, and we do know that the virus moves with people, and this has only just confirmed that even more. And almost every country in this list has now reported cases of B117. What I think would be nice to show next is more recent flight data and to compare how movements of people have changed and how the dynamics of that has shifted a lot from October to, say, December, and even more recently when actually there's probably been a significantly less amount of international travel. So we're also tracking media reports of either imports or local transmission to try and counteract the lag of sequences getting onto GISAID, or maybe lack of sequences getting onto GISAID as well in some cases. And this number is always a little bit higher. There's 114 different countries reporting cases of B117 around the world now, and local transmissions being reported in various parts of the world. How many? The thing is, though, there's still a lot of biases associated with this. A lot of countries are only sequencing travelers, so we may not have any idea of what's actually circulating in the community. In general, a positive note is that we've seen most countries do closely follow a report of a lineage of concern with upload to GISAID, which is great because it means that we can continue to track the lineages by sequence count, which can be done automatically through Grinch. I won't go into a huge amount of detail for time, but we've also been tracking lineage B1351, which was first identified in South Africa. And to date, there's 67 different countries reporting cases of this lineage. We have flight data from them too, but one thing to note is that this flight data may not be quite as representative of the ultimate movement of people. So we've got access to the destination on a single booking, which may not be final destination. We've got Ethiopia and the UAE as the top 2 destinations here, which are hubs for onward connecting flights. Also, South Africa is not an island, so there's likely a significant amount of overland movement that we're not accounting for in this graph. And we do know that neighbouring countries and a lot of countries in Southern Africa do have quite high prevalence of B1351 now, and that's not represented necessarily in this figure. Slightly after we first started, we began tracking lineage P1 then as well, and we've now got 36 different countries reporting cases of this lineage worldwide. Comparing the same, we've got flight data specifically from Manaus to destinations around the world, and the picture has changed a lot since January, which is when we first started tracking P1, and today where again, we're seeing this confirmation that viruses move with people movements. So just to summarize, Grinch is quite a simple but effective tool that allows us to produce these daily reports and keep on top of where the virus is circulating at any given moment, and these lineages of concern. It uses the assignments that it produces anyway from Pangolin to also maintain the individual lineages pages for all lineages on covlineages.org. So that website is now being maintained and updated on a daily basis, thanks to Grinch. And it's recently had a code refactor, so it's flexible and can be scaled and track new lineages of interest, and can be added to get this suite of reports, which it does automatically now. And I won't mention them today, but we're adding new features to it as well that can help track constellations of mutations, and that will be mediated as well by Grinch. So it's doing a lot of work, and we've got more to come as well. Just to give some particular thanks to everyone in the Rambo lab and anyone who contributed to the reporting tool, particularly Verity and Moritz. So thank you.
Thanks, Áine. That was really good, and it's a very useful resource. A lot of thanks. So do we have some questions? Yeah, the first one in the chat. What are the implications for travel to and from the UK? And then the second part, are other countries increasingly sharing their data?
So, okay, the first part, you can see this huge confirmation of when we only first started tracking these variants of concern, and now you see that the virus moves with people. It may take us some time to actually detect that and to sequence that, but it does happen. And so I think the figures sort of speak for themselves in that case. And the second part was, sorry, can you remind me?
Are other countries increasing their sharing their data? I guess.
Yeah. So other countries have, honestly, it's been a huge global effort. I checked recently, and there's, I think, 166 countries now around the world with sequences of data on GISAID. And when we compare when a media report goes out, most countries closely follow that with sharing of data. And I think it's very rare now that hasn't been the case. I think since the very beginning of this pandemic, actually, there's been a huge push for real-time sharing of data, which has really enabled us to track lineages like this.
I think it's fair to characterize what happened at the end of last year as a sort of second phase. That's sort of been starting to creep in, this idea that there's a shift from a first year till sort of now.
I think there was a lot of build up towards the end of 2020 that this all may have been behind us, but it does seem to have escalated, and we do seem to have reached a turning point. And it's not just these 3 lineages of concern. We do see quite a lot of new variants coming up kind of in a global concerted way at the moment. The dynamics of that are very interesting. Yeah.
Thank you. That was really interesting. Thank you. And so the next speaker is Matt Sinnott from the Wellcome Sanger Institute. He's going to talk about SARS-CoV-2 mutation and variant surveillance, and that's been performed there.
Great, thank you very much. There you go, just waiting for the Zoom banner to go so I can actually share the screen. There we go, can you see the slide? Is that all good? Yes, okay. Okay, great. Great. Okay, well, thank you very much for having me. It's a real pleasure to be speaking with you today alongside such an excellent lineup. So my name is Matthew Sinnott. I am a genomic surveillance data analyst here at the Wellcome Sanger Institute. And today I'll be talking with you about the work we've been doing on mutation and variant analysis of SARS-CoV-2. So Sanger is currently sequencing more than 2,000 samples a day and uploading them to client to aid data analysis across the consortium. And Cordelia Langford was talking a bit about that process in previous talk. So, but alongside this, we want to try and use this wealth of data we're generating to generally answer a couple of broad questions. What information can we provide for surveillance, and which of the sequences we are generating represent a particular concern? So the main aims of the data analysis team are to rapidly call variants, to communicate rapidly the discovery of variants of concern, and also just general surveillance of the pandemic across the country. When I talk about general surveillance, that refers very much to analyses that utilize a randomized sampling of viral genomes from across the country. And doing this allows us to avoid biasing analyses based on location. Now, the main reason I bring this up is that the recent rise of concerning new variants has led to more calls for variant surge testing. So variant surge testing is where in a particular area, if a variant of concern is seen, the amount of testing in a particular area may be increased and more of those sequences may be sampled. More of those samples may be sequenced, other way around. And Sanger has been asked to contribute more to the sequencing of these surge tests. So this caused a couple of sort of main complications. The first one being that it required fast adaptation of the existing pipeline. And Cordelia Langford again talked a bit more about the sort of turnaround times involved in this. This is just a little depiction of how a lot of these processes had to be really sort of sped up. So that was one complication. The second complication is that it is very important to separate out these surge samples from general surveillance samples. And the reason goes back to what I said earlier about needing to make sure that we're not biasing towards a particular location. So it was very important that we have surge and surveillance samples labeled and separated out so that different analyses can be done on each of those. So to aid the increased rapidity of the platform, the rapid variant of concern calling platform was born. So this involves two main steps. First of all, we perform rapid variant of concern genotyping, and this uses variant definitions from Professor Nick Lohman and Public Health England. And then asynchronously to that, we run Pangolin. So this will basically provide lineage designations to all of the samples, not just those that are genotyped as variants of concern. So this gives us a pipeline which will first of all detect variants of concern as a priority so that we can report those rapidly to public health agencies before then going on to label other lineages and providing that data for both internal analyses and for analyses across the consortium. So now that I've talked a bit about the sort of data pipeline, I want to now talk about a couple of applications we've been developing to use some of this data. So back in December, we produced a virological post which used a Bayesian inference model developed by Harold Vohringer and Moritz Gustung at the EBI. And in this post, we presented that over the November lockdown, other viral lineages had been contained, whereas B117 had escaped containment. So the work in this post has now progressed, and I now want to present a web app that went live literally yesterday. So this is very new. It's at COVID-19.sanger.uk. Please do visit. So as you can see, we basically present a choropleth map currently colored by the proportion of B117 on the 1st of December, and we provide these interactive plots for incidence, proportion, and R rate. So we can zoom in on a particular area. So we'll go to Swale, where we first saw the rise of B117, and we can see that these interactive plots change to show these factors for that local authority only. And so we can just go back to the beginning of the time series, and then we can show the progression of this of the B117 across England. And so you can see how it starts down in Kent. And then we can color it differently. So we can color by R rate. In the first version, we show it with three potential lineages. So we show B117, B177, and then we group all other lineages together. For later editions may include more lineages. So, and we can change the coloring as well between linear and quadratic. So this gives us our lineage-specific spatiotemporal tracking, as I say, based on Bayesian inference model developed by Harold and Moritz, and the lineage information is provided by Sanger, but put together using Pangolin. So that's the sort of new web app that we have recently launched. Now I would like to go into a different application, which is more sort of in progress in development, which is the super spread search. So a super spread event is one where a single person may transmit the virus to multiple people at once. And this can quite often seed a much larger outbreak. And so what we want to try and do is create some sort of early warning system, if you will. So how do we try and identify these? So I've given sort of two examples. If you imagine that each of these dots is a viral genome. So in the left-hand box, we see what we might expect to see with diffuse community transmission. So we have lots of different viral lineages, none of them really overtaking the genotypic background. Whereas in the right-hand box, this is more what we'd expect to see in terms of a super spread state. So we would see this red genotype is dominant across the population. And so we would say that it's likely that all of these arose from the same event. And therefore we might say that this may well be a super spread event to worry about. So to quantify this, we employ something which we developed called homozygosity statistic, focused on sort of population homozygosity. So that we define this as basically the probability of pulling two identical genomes with a little bit of wiggle room out of a given population, where that population is are the sequence samples in a local authority within a given 7-day period. So as a sort of case study of this, I want to sort of bring you to Swindon. So in July, there were reports of a potential super spreading event at a food processing factory. And now on around the 16th of July, we started to see an increase in our super spread statistic. And this was followed a little while later by a rapid increase in cases in Swindon at its peak, reaching around 5 times the national average at the time. So the fact that we got a sort of signal slightly before this rapid increase in cases may suggest some sort a use case for trying to predict where we may want to sort of look at a bit more closely and maybe look at employing more sort of national restrictions. Sorry, local restrictions, I mean. So how would we'd want to sort of present this information? We would like to present this first of all as a sort of national overview, where we'd say color areas in yellow or red, depending on whether we believe that they are mild or high risk. And then we'd also provide something like a local report that may look something like this. So we would provide a timeline with showing how the statistic has performed over time. We may show a density plot that shows where that particular local authority is in comparison to the rest of the country at that time. And also we would try and show as well neighboring regions and try and show how they're faring in terms of this statistic as well. So that's in a broad picture, that's sort of how we see this panning out in terms of looking for super spread events. Now, something which is more of a a sort of recent thought and like a sort of something to look at in the future is the potential for this to inform early on where we may be seeing a new variant breaking out from the background of other lineages or variants. So basically, I'm going to bring you to Kent, which, as I'm sure we pretty much all know, back in November, we were seeing a rise in cases even during the November lockdown, primarily because of the rise of this new, more transmissible variant, B117. Now, as I'm sort of showing in this plot, alongside that, we had a rise in the Kent homozygosity rate compared to the rest of the country. So in November, at its peak, it reached around about 3 times the national average for homozygosity. But then once restrictions were released, we got another very large spike in this homozygosity rate, reaching around about 6.5 times the national average at the time. And that then led to, at the same time, we started to see a much larger increase in the rate of cases going up as well. So now this is very much a retrospective view, but the sort of thought is that possibly with sort of future new variants, seeing a pattern like this may give us an early indication that a particular rise in cases is not necessarily just due to larger community transmission, and it's not necessarily just due to, you know, sort of people ignoring the rules in a particular area. It may be, so it may give us more of an indication that there's an underlying biological cause, which in this case, we know, we are pretty certain that the underlying cause here was the rise of this new variant B117. So that's what I wanted to talk to you today about in terms of our applications. As Cordelia mentioned before, none of this gets done without an incredibly large and dedicated team behind it. So we want to pay tribute to everybody in the Sanger COVID team for all of their efforts in this. I've picked out a few names on the side here who we've been working with a lot on the data analysis side. So many thanks to Jeff, Dominic, and also a special thanks to everybody who got the web app running off the ground quite recently. So thanks to Twee, Theo, Sam, Richard, Harold, and Moritz for all their help with that. We also want to thank the Department of Health and Social Care, Public Health England, the Wellcome Trust for their support, and thanks as well to everybody in COG-UK for all the developments, which have also made it much easier for us to work on our tools as well, and you've been absolutely instrumental in that with us as well. So thank you very much for listening, and a reminder to please go and visit our new web app at covid19.sanger.uk. Thank you very much.
Thank you, Matt. That was really interesting and a very impressive amount of teamwork going into that. A few questions. We don't have a huge amount of time. What's the super spreader wiggle room SNP cutoff?
Yeah, so this is actually something which is sort of a little bit of flux in terms of the methodology, and is likely to sort of change. So currently we use the output. We use filer type designations that are produced using filer pipe. So the filer type basically provides us with a string representation of the position of the genome on the phylogenetic tree. We basically the wiggle room that we talk about is basically allowing for a particular genome can be matched with another genome that is two mutations behind, basically. So two SNPs behind, basically. So that's the wiggle room we provide at the moment. As I say, we are sort of this is something that is likely to change in order for us to try and speed up our processes as well, and we're sort of moving we're likely to move methodologies somewhat, but that's the sort of current implementation anyway. And
so somebody's asked, is it possible to share this to look at another country, or how would, if somebody was interested, is the tool, is there any plan to develop the tool further, I guess is the question. Is it in that in terms of the super spread tool? I think more the dashboard where you were showing the lineage. Dashboard. Yeah, I mean, so at the moment, it's very much for the like basically to serve the model which was developed by Harold in terms of the UK. It's very reliant on you know, sort of wide-ranging genomic surveillance. So that's why it's sort of, you know, it's very well tuned to the amount of surveillance currently occurring in the UK. But you know, if with robust enough surveillance, it probably could be tuned to other countries as well. So, but that's not really something I can answer myself. It's much more to the model people. So Harold and Moritz might be able to shed a bit more light on that. Thank you. That was a great talk. There's a few more questions in the chat. I think you should be able to see those, hopefully, and you can. I'll have a look and try and reply to some in the chat. But yes, thank you very much. And so our next speaker is Alessandro Carabelli from the University of Cambridge, and he's going to talk specifically about prioritizing mutations and variants.
Thank you, David. Can you confirm you can see my slides, please? Yep, see them. That's great. Okay, so thank you. And thanks, COG, for the organization of this showcase. And it's an honor for me to contribute today. So today, essentially, I'd like to share some of our thoughts that we believe are important if we want to guide experimental work and better understand SARS-CoV-2 evolution. So this is actually something which is a matter of discussion among many of us at COG, and it involves more directly many partners like Public Health England and Hygiene Consortium. So these are some visualization from Next Strain to look at the development of the pandemic. These are all unrooted trees. The branch length shows the divergence, and the background aligns on current data. And the circles on these trees correspond to what was available last year on the 17th of March, in October, and now. So at the beginning of the pandemic, all mutations were accumulating at a relatively very slow rate. We heard many times this magic number of 2 mutations per month in the global lineages. And generally, early on in the pandemic, the virus was evolving steadily into humans, and it didn't need to change much because of our susceptibility and its high transmissibility. And then at the end of last year, there was the emergence of the variants of concern. And as said before, so much changes at once, as you can see from this branch for the B117, was quite surprising. So there is a big question about SARS-CoV-2 evolution. We actually don't know the answer, but what we know is that we can actually start structuring how we think about maybe experimental work. And this is actually the aim of prioritizing mutations, because what we want to do is to understand the future of the virus and what comes next. So if we talk about priority, we should start asking ourselves, what are the classes of important mutations we should focus on? So this, we think can be broken down into 4 categories, what we call TILT, acronym, which I've stolen from Sharon. Under our attention, there are sites known to change virus infectivity and transmissibility. There are mutations with immunological roles, either affecting monoclonal antibodies, vaccines, T cells, in general, the host immune response, or because associated with increased virulence, so therefore changing the clinical landscape. And finally, regions affecting primers, probes, binding sites, which are very important in the diagnostic, as we heard earlier. So when we looked at some of the mutations, it was clear early on there were going to be some sort of antigenic changes, especially in the spike protein. So this is taken from a beautiful work led by Emma Thompson and David Robertson. It's a story about N439K spike mutation. So this mutation is one of the first examples of antigenic changes in SARS-CoV-2. And so from structural analysis, they found that the virus, through this mutation, was changing the interaction with the human ACE2 receptor and possibly escape some monoclonal antibodies and to a less extent some convalescent plasma. So this was telling us that there is a lot of plasticity in the spike protein. So the spike accommodates changes in the conformation and can alter the interaction with the ACE2 receptor. So the virus clearly tolerates mutations in the spike gene. As you can see from this graph in the middle, showing the different amino acids of the spike protein and the frequency of their mutations. So this shows that many mutations are occurring at almost any amino acid of the spike. But if you look at the spike here on the right side, so in the top view and side view here with these videos, in false color, you can see the antibody footprints, human antibody footprints, which are classified here in 5 different classes. So as you can see, most of these antibodies are targeting the same top region, and the region here in purple a bit more lateral within the N-terminal domain. So that's what is very interesting to think about in terms of what we are predicting as variants of concern, because one of the possible worst case scenarios would be a variant that can carry mutations that can abrogate all classes of antibodies. So I wouldn't go into many details here, but the key message of these slides is that we started looking at single mutations, and by plotting the frequency of those mutations, we can nicely see waves of variants in the UK that had different reasons for spreading, either driven by fitness or possibly by demographic reasons, as in the case of H2V, for which apparently last summer travels played an important role. However, soon we realized that there is a lot of mutations that we were looking. So what's happened is that the number of important mutations has really increased remarkably. So this is a screenshot of several variants of interest that are circulating in the UK, and Public Health England is doing an amazing job in horizon scanning. This table is from the COG Mutational Explorer. COG-Me is one of the several tools which are available at COG, and you probably saw the link to the website already in the chat box. Right? Anyway, this is the link if you want to have more information. So despite this increasing number of different mutations and variants of interest, so as we heard earlier, interestingly, a lot of convergent evolution is going on with a lot of same or similar mutations popping up in different lineages. So these lineages are not static and dynamically can acquire new mutations that can potentially affect the entire phenotype of the variant, as in the case of the E484K appearing in the B117 UK lineage. So this figure shows the spike protein of 3 variants of concern and the MINC associated cluster phi. In red, you can see the position of lineage defining mutations. So in this we show several properties. It's quite complex. I'm going to try to guide you through. Here, for example, by using bioinformatic tools, we can see how accessible to antibodies a specific site is, whether a specific site belongs to a footprint of any class of antibodies, or whether there is an experimental evidence of any antigenic change here in black, or any change in the biological properties like the ACE2 binding affinity. So it's quite nice here how we can visualize how a combination of mutations, like for example the 501Y and 417T in the lineage P1, and or the K417N in the B1351 lineage, have opposite effect on binding affinity, potentially compensating significant structural changes that are occurring on the spike. So looking at the mutations that define all those specific lineages, it's clear how only a subset of these mutations are going to be phenotypically relevant. And it's also clear there are like different functional domains of properties, binding affinity, antigenic changes. Another one is the furin cleavage sites here, shown with a pair of scissors, which is, I'm not going to discuss here in details, but that we know it's important in the cleavage of the 2 spike subunits, and it's very important in cell entry. But what we actually don't know is how all these mutations work together, and if they are all linked to other mutations outside the spike, which is very important if you think about the prioritization. But we definitely need to map out what is occurring now against what is not observed yet. So pragmatically, what we are trying to do is to bring together evolutionary understanding, selection analysis, structure context, frequency, experimental evidence coming from the literature, which is exploding, by the way, and bring it all together in order to understand where the virus is going forward. But surely in the context of prediction, what matters the most at the moment is whether there will be the insurgence of a new variant they might escape immunity, both generated with previous infections or through vaccination. There's a great risk of importations or exportation of variants of concerns, like for example of a new variant identified yesterday in 3 samples in Angola, which carries 31 amino acids and amino acid substitution and many VOX-like mutation. But we also believe that we should be concerned of any additional change accumulated on the top of B117, which is what this heat map shows in the right side. Here we show frequency of antigenic changes on the top of B117 in green over time. And we know from the literature that some of them have the potentiality to affect either monoclonal antibodies, convalescent plasma, and vaccine, here in black. We are definitely worried about the insurgence of a new variant that might carry mutations that can affect all classes of antibodies. Although in the context of fitness, over the last few months, these mutations might not have been very advantageous, we don't know whether they might become very advantageous in the near future, in particular in the heavily vaccinated population. So to conclude, we observed a dramatic shift in SARS-CoV-2 variants towards the end of 2020. The VOX seems to have a toolkit where they can accommodate functional domain changes. They can accommodate conformational changes, increase binding affinity, have antigenic changes, etc. But how do they work together, all these mutations? This is a kind of questions that we have to answer through experiments. And while vaccines seems to be working, we need to continue monitoring their efficacy as much as about monitoring changes to already circulating variants. And these changes are already happening, in particular on B117. And we need to find out how several new set of mutations will affect immune efficacy, and in particular, if they will affect all classes of antibodies, or in general, the immune response. If you think about T cells or the innate immune response. And yes, there is a lot of people to thank on this, in particular David and Will, Derek and Joseph, who helped with the dashboard, the COG-Me, and all the ones that are more directly involved with the mutation analysis working group at COG. And thank you very much. I think I will stop here.
Thanks, Alessandro. That was a really good talk. I think we have a few questions. Two related questions. Is machine learning helping with any aspect of the variant work to date in predictive manner?
So at the moment, we are not using any machine learning. So this is something definitely we should think about. But
I guess that leads to the next question. Is it possible to predict new mutations?
So I think this is a very interesting question. I wish I could answer yes, but I don't think we are at that point right now. What we can do now is definitely think about any possible scenario, any possibilities. We start now kind of identifying these functional domains, like a sort of toolkit that the virus can use. And that's the aim of my presentation for today, was more like showing like possible tools about the virus and how we should kind of plan our experimental work and trying to figure out how all these tools are brought together by the virus and used and to change its phenotype.
And there's one question here. There are some concerns that mass vaccination based on the spike protein might increase new variants or generate mutations. Do you think that's something that's justified? I guess vaccination versus high infection rates. What do you think?
Yes, I think that definitely with the vaccination, the virus would receive a sort of evolutionary pressure, and so we expect that since we've seen that the spike, for example, is extremely plastic, so there is a sort of plasticity. We might see any sort of mutations that might be able to escape the immune response boosted by vaccination. So this is something that we are kind of waiting. And,
thank you. We better stop there. There's a few minutes then, a break before we start at 2:45. Thank you, Alessandro. That was really good. Good talk. Thank you.
Thank you.
The next session will start in 2 minutes. Thank you all.
Good afternoon, everybody. My name's Sally Corden. I'm chairing the last session. Real privilege to be part of such a fantastic afternoon of talks, and the last few talks I'm sure will generate just as much interest, if not more than the previous ones. So without further ado, I'd like to introduce to you Professor Judy Breuer, who's going to talk to you about the HOCI study. Thank you, Judy.
Thanks very much, Sally. I'll just share my screen. I can just see the presentation. Okay. Can you see that?
It's just come through, Judy.
Okay, so thank you very much for this invitation. I'm going to tell you about the HOCI onset hospital in the COVID infection trial. So what is the oh sorry, is this, here we are. This is my first slide. Sorry, the first slide is entitled "What can we learn from HOCI?" So HOCI is a phase 3 prospective interventional cohort superiority study. It's urgent public health supported, and it's asking basically two questions. Does the availability of SARS-CoV-2 sequence data to infection prevention and control teams, first of all, reduce the incidence of hospital-acquired SARS-CoV-2 infection and reduce the numbers and sizes of hospital outbreaks? And
Judy, can you just put it onto slide view for me, please, so that people can see it?
Yeah, I'm sorry, I don't know how to put it on because it's on Zoom on mine. It's on the screen share on mine, but it just doesn't seem to be translating to screen share on the screen. So anyway, okay, so basically, and then is the impact of sequencing on IPC practice dependent on the time to result? So essentially, this is about sequence data in the context of hospital infection. And we've heard a lot about how important the COG-UK sequencing was for public health. And I think that is now, without any doubt, accepted. But we still have some questions about where it will fit into hospital outbreaks and how we'll be able to use it. So the methods we used were to actually recruit 14 sites across the country, and they had a different sampling policy from the rest of COG-UK, and they sequenced all SARS-CoV-2 positive patients, inpatients, and healthcare workers. And that's going to end up with around 30,000 patients and around 2,000 patients who've acquired the infection in hospital. So it's no selective sequencing, everybody was sequenced. And the sequence data, patient metadata, and community sequences were all integrated into a probability model to try and identify the HOCI infections and the linked infections in a simple report form. We compared baseline data on doing no sequencing where possible with rapid turnaround sequencing and then a period of slow sequencing. And then we have also got a qualitative evaluation of why things have worked or not, and also a health economic study. So the study commenced in October 2020. You can see this is where it was in relation to the national picture, and we were just before the B117 came in, and you can see the rise in cases, then the slight diminution before the B117 took off. And the grey area represents the HOCI study, and you can see that we haven't finished, so I haven't got the final results to report. We're finishing at the end of April. This is study recruitment so far. You can see that in relation to the second lockdown and the third lockdown, and also the Christmas closure, which was a problem for most of the sequencing centres because they were university centres, although to be fair, a lot of them did continue to sequence. And you can see that we sequenced the majority of cases after Christmas, and the baseline phase was generally up to about here, and then rapid and slow phases after Christmas. So you can see that so far we have recruited about 1,500 cases, and you can see that they are both at baseline and during the rapid sequencing, and that there are fewer in the slow sequencing, but this was shorter. This was only 4 weeks, and baseline and rapid sequencing were 8 weeks each. And we have a spread of cases who've acquired the infection at different times following admission to hospital, and roughly the same proportions before and in each of the phases. Just of interest, you can see here that this is a plot of the ages, the age distributions, or the numbers of people by different ages during the baseline phase, rapid and slow. And I told you the rapid and slow came after Christmas, and you can see that the age has shifted and there are fewer people over 70, and we're wondering if this is an effect of vaccination, and that's something we will have data on and we can look at. So what else? So we're going to learn a lot about the use of sequencing in managing hospital infections, but what else can we learn? Well, one of the things that has come out of this very strongly is that we need to have better tools to help with the interpretation of sequence data in hospitals. At the moment, we all tend to use the phylogenetic trees, and this is a very nice CIVIT report, but this is not something that infection control teams find easy to get their head around, and it can be complicated for other people as well. So our aim in this study was to actually develop something that allowed us to give a single page report on a patient who acquired the infection. It identified those cases that were highly likely to be linked to the focus patient, and it provided a schematic of where the patient, lozenge shape, was in relation to linked cases, in order to provide the infection control team with a quick snapshot of where they should be focusing their attention. And HOCI will test the utility and acceptability of this tool in managing outbreaks and identify how and where it can be improved. So what else can we learn from HOCI? Well, we also had the opportunity to look at variants, and particularly the B117 variant. And in this study, we had 9 of the sites. This actually should be on an animation, so I will move it out of the way for the moment and then move it back again. Oh dear. So basically, what this shows is a plot of the relationship of the association of B117 versus non-B117 with mortality. And you can see here that several studies, and you'll hear a bit more from Nick Davies after my talk, have shown that B117 was associated with an increased mortality. These studies tended to all use the S gene target failure, and that's shown by the sort of yellowish color. They did not use the sequencing, so that is a potential bias, although that was corrected for. And they also tested, most of them tested, used samples that were tested in the community. The only ones that have used samples that were tested in hospital is the HOCI sub-study. And you can see here that we found much less association with mortality overall, although we did find this interesting increase in association of B117 with mortality in women, and we're looking into that further. To be fair, other studies that used community tested samples for S gene target failure also found that hospitalized patients did not show an increase in mortality. So we will be the largest study of patients who've been tested in hospital, and we know that only about 10 to 15% of people who are tested in hospital have a test in the community beforehand. So it's possible that the results from these, although very interesting, may not necessarily reflect the results of the hospitalized patient population. And similarly, when you look at ITU admission as a measure of severity, you can see here again that there are far fewer looking at this. Again, there was a primary care test, cohort that was followed up for admission to ITU, tested by S gene target failure in the community. This Danish cohort used the sequencing to test. Both of them showed an increase in mortality. And our own study and a small study by UCLH, which did use the lineage data tested in patients who are already admitted to hospital, does not show an increase in mortality in admission to ITU. Although again, we have this strange result for women, and we're looking into that. We've also been able to use the HOCI study to look at the impact of lineages on hospital transmission. And you can't see this, I expect, very well, but what this shows is that the B117, the risk of B117 was actually lower in patients who acquired the infection in hospital, suggesting that it was not responsible for any increase in nosocomial transmission. So what will we learn overall from the HOCI study? Well, we'll learn about the benefits and needs or not for rapid pathogen sequencing for infection prevention and control in hospitals. We'll learn about the factors that affect the delivery of that sequencing in NHS hospitals and how to improve this. That will include things like proximity to site, how well the sites coped, the sequencing sites coped. We will also learn about the factors affecting the use of pathogen sequencing for infection prevention control and how to improve these. And this will include how we can improve on the tools that we will need to have if this is going to be widespread, costs, nosocomial spread, and this will form the basis for discussions of how Pillar 1 sequencing will fit into UK future plans, which at the moment is not necessarily being thought about in terms of patient management, and this will hopefully inform that. And we will also, from this study, learn about the relationship between lineages and disease severity and nosocomial spread. An exciting development is that we are linking this whole data set to through ISARIC and through COG-UK to patient NHS numbers, and that will enable this data set to be used for studies to look at things like lineage and viral load in hospitalized patients. We will have the HOCI patients who have a defined date of onset. So that is quite an interesting way of looking at viral load. We will have the relationship or none between lineage and nosocomial transmission, lineages and outcomes. And we will also be able to link this to more data on patient severity and patient clinical outcomes. And indeed, we will be able to link it to the host genomics on patients who are also entered into ISARIC. So there'll be, it's a particularly unique sampling strategy within COG-UK, and we hope that we'll have lots of utility beyond just the study. And I'd just like to finish by acknowledging all the sites, the 14 sites who contributed to HOCI,
and all the people at UCL and the Wellcome and COG-UK who contributed to the setting up of HOCI and its success. Thank you very much.
Thank you, Judy. One question that's come through is what assays were used for the rapid sequencing and how quick was rapid?
So that's a very good question. The assays varied. We didn't prescribe assays. So some people used ONT, other people used Illumina, and most people used the Arctic amplicons or the Illumina amplicons, whichever ones you had access to. But we didn't prescribe the platform. And rapid, it varied, but most people were able to achieve 48 hours or less sequencing turnaround, barring things like the occasional contaminated plate and that kind of thing. The issue really has been in the sample transport.
Thank you, Judy. And one quick last question. Are you looking at intrahost mutation in hospitalised patients, particularly those with prolonged infection or immunocompromised?
So we're not looking at that in this study, but we have, as a nested study within this, there is with a couple of the sites, we are actually looking at that, yes. But we're doing that as a separate investigation with the sites that we provided sequencing for.
And one very quick question for myself is, are you collecting any user feedback on your report?
Completely, absolutely. That's a really big part of it. We've got a whole quality assurance assay, and people are filling in forms and talking to the health quality team, and they're doing interviews with everybody and including the team that used the report. We want to find out how people find it and what would be needed to make it better.
Thank you, Judy. Look forward to hearing when you have the final reports ready. Okay, we're going to move on now to Dr. Nick Davies. Yeah, thank you. So we'll just have a switch then. So Wendy, I hope you're ready. Professor Wendy Barclay is going to talk about the SARS-CoV-2 variants, vaccines and immune escape. So hope you're ready, Wendy.
I will attempt to share my screen and see if I can get there.
Friday afternoon gremlins, I think.
I'll just try and get that onto that way. Slideshow.
Thanks, Wendy. If you're able to go onto slide view, I know that Judy had problems with that. Brilliant. Thank you very much.
Okay, thanks very much. I must thank the whole of COG-UK for a fantastic afternoon, and special congratulations to Sharon. I'm here really representing a different consortium called G2P UK, which is much younger. Our birthday will be in February next year, so we're much younger than COG and have much to learn from you. So thank you very much for having me. G2P stands for genotype to phenotype, and we were conceived with an aim of trying to take all the wonderful information that's coming through from the sequencing efforts that we've heard about today, and to try and interpret some of that sequence information in much the way that Áine was beginning to describe. But really the G2P Consortium are a group of UK virologists who largely are wet lab virologists who can perform in laboratory studies in vitro and in vivo models. And what I'm going to try and do today is to use illustrations from some of the work that's beginning to come out from the group and also from other laboratories to show how that kind of approach can complement the sort of genomic approach that COG-UK take and begin to help us understand that translation of genotype to phenotype, which mutations matter. So I think we're all fed up with this. It's been a long haul and it's not finished yet. There's been social distancing, non-pharmaceutical interventions, and we're basically hoping that vaccines are going to be the way out. And we've seen this fantastic accelerated vaccine response where from, in less than a year, we have vaccines in people's arms. And we're all familiar with the kind of data I'm showing here. On the left, this particular one is the Pfizer results that was published in the New England Journal. And basically what that illustrates is that by immunizing people against the spike protein, We induce antibodies that bind to various parts of spike, including the receptor binding domain, after a single dose, and then lots more antibodies after a double dose. And that appears to correlate to some extent with protection from severe disease, infection, and transmission of the virus. However, as we've heard throughout today, SARS-CoV-2 genome is mutating, and that is so well described by this consortium. And we've heard a lot about constellations of mutations that are being picked up in variants that emerge, and how important it is to think about the constellations. But some of the mutations within those constellations might be hitchhikers. Some of them may have little consequence, whereas others may be actually driving the phenotype. And what we need to understand is which are which. Which of the mutations here might be, for example, conferring escape from immune responses, importantly those vaccines, because that's going to be a fly in the ointment for our way out. Of course, we may also have mutations that change the behavior of the virus in terms of increasing transmissibility or affecting the disease it causes. That might affect our treatment options. And there's a worry at the moment, possibly about some of the mutations increasing the opportunities for crossing back into animal reservoirs, which may, in the long term, make it much more difficult for us to control this virus. So it's a critical time, as Alessandro pointed out, since sort of the late 2020 period, we've seen this appearance of mutations, possibly because more and more people have been immune, but possibly also because we changed our behavior, or possibly because the virus had circulated in the human host for a while and then had the opportunity to change. So how do we stand at the moment? Well, there are three variants of concern, which I will mention today. The UK variant, B117, the South African variant, B1351, and the Brazil variants, which we tend to call P1 and P2. And I'm going to go and talk about each of these through the talk, but as has been noted, I think one remarkable thing to keep in mind is that there is a level of convergent evolution being illustrated by these variants as they arise in different parts of the world. So to turn to B117, as we've heard today, it was detected in the United Kingdom in the southeastern area, but of course has spread very widely throughout the world, as Áine showed us, defined by lineage-defining mutations that span the whole genome, but much of the work so far has concentrated on the mutations which sit in the spike. And as we saw, rapid growth, suggesting a biological switch conferred by those mutations, which might be conferring increased transmission. If we think about transmission then, what does that mean for SARS-CoV-2? Well, we can break transmission down into somebody being infected, virus accumulating in their respiratory tract, virus being emitted into the environment in airborne droplets and also deposited on fomites and shared surfaces, and then virus finding its way into the target tissue of an exposed individual. And if we think about how mutations, for example, in B117 might affect the efficiency of that process, they could work in many ways. They could result in higher amount of virus replication, a longer duration of shedding, for example, survival in the environment, or perhaps altering the infectious dose required to initiate infection upon exposure, which could be a combination of changes in the way the virus sees its receptor or evades the innate response. And then, of course, there could be things that alter the number of susceptible individuals. The virus might be evading the acquired immune response, the antibody response of people already infected or vaccinated, or perhaps able to infect and transmit between different groups of people that it was not infecting before so much. So where are we at the moment? We're trying to apply our phenotypic characterizations in the laboratory to try and understand that. Well, I think what we've learned in the last few months really is that if we take isolates of SARS-CoV-2 variants and we do our best assays in the laboratory, and this is a growth curve in primary airway cultures of human airway epithelial. So this is about as close as you can get in the laboratory to a controlled inoculation experiment comparing 3 different variants, and the blue one here is the B117 that we know is transmitting well in the community. You see that we see no statistically significant difference in the growth. So our lab assays are not picking that up, that difference that we see in real life. However, a very nice post earlier this week from a French group pointed out that if you actually perform these assays as a competition, where you inoculate a wild type virus and the B117 together at different input ratios, and then sequence after several days which of the 2 genotypes emerges predominantly, then every time it is B117. So I think what this tells us is that there is a biological difference in the replication capability of this mutant or this variant in airway cells, but it's subtle and you have to work pretty hard in the laboratory to find it, and that we need to employ these kinds of competition assays going forwards to do so. Why might B117 grow better in human airways? Well, as Alessandra referred to, we can see important sequence changes in the receptor binding domain, particularly at position 501 of B117, that likely enhance its affinity for ACE2. Here you can see that in close up, this 501 mutation at the top of the receptor binding domain of the spike protein interacting with the ACE2 receptor. Not only that, Alessandra also referred to a furin cleavage site, which is important to help the virus enter cells. And here we've used either expressed spike with different mutations as pseudoviruses or authentic viruses, and measured the efficiency of cleavage of the B117 versus the wild type virus. And we see small but significant increases in cleavability of furin cleavage site, which translate into small but significant increases in the ability of the particles to enter cells as measured in this pseudovirus entry assay. And that again maps to this mutation close to the furin cleavage site. And that taken together may also impact, for example, the susceptibility of this virus to naturally induced interferon. So we know SARS-CoV-2 is susceptible to interferon, and probably that's because some of the interferon stimulated genes that get activated when the interferon system is kicked in as the virus is recognized, some of those genes act on the virus directly and stop its infection. Some of those may well be, for example, IFIT-M proteins, which are known to be pretty good at sitting in the endosomes as viruses enter and blocking their uncoating. On the other hand, a virus which can use furin very efficiently to cleave itself and then enter through a non-endosomal pathway, as SARS-CoV-2 does, can escape perhaps the effect of those interferon-stimulated genes. And indeed, this is a preprint again from this week from a Spanish group who show that the B117 variant is less susceptible to interferon than the previous virus. So what I'm hoping to show you is that using combinations of assays in the laboratory, we can begin to break apart, if you like, the biology of these variants and understand how they're working and why they're having the phenotypes that they have. And the most important one really at the moment, as I said at the beginning, is really this business of, well, do these variants escape the antibody response? Because that's what we're looking for to help us control the virus in this next phase as we go forward and vaccinate a lot of people. And the good news is here that the B117 variant, which of course is predominant in the UK, does not escape antibody neutralization to any significant degree in convalescent or vaccinee sera. And you can see that, for example, here in a bunch of convalescent sera in work that we pre-printed, and then here also in work from the CRIC. The difference between the neutralization titers of old virus and B117 are twofold at the greatest. On the other hand, what you'll spot on the end here, and what is well known, of course, is that this South African variant B1351 is showing a much bigger difference. So that lack of difference in the laboratory for neutralization of B117 thankfully converts into real-world vaccine effectiveness, which is very good in the UK. A number of different reports now from Public Health England and Scottish Public Health illustrate how our vaccines are working, even in the face of the B117 UK variant. On the other hand, of course, as you're aware, the South African variant B1351 has arisen in a different part of the world at around about the same time as our B117, but here we have much less confidence about that antigenic distance. In other words, this variant arose in an area of the world where there were high numbers of infections in the first wave, so presumably high seropositivity at the time it was emerging. And indeed, there are worrying signs, for example, that people who were in the placebo arm of one of the clinical vaccine trials had a similar infection rate, even if they were seropositive at the start of the trial, as those who were seronegative. A follow-up suggests that slightly less drastic result, but nonetheless, this is causing some concern. And then, of course, we have Brazilian variants, P1 and P2, which also emerged in a part of the world that had a very high first wave, 70% or more seropositivity as this virus was emerging. I'm going to skip through that and remind you that the key mutations here then in the South African variant are, and the Brazilian variants are positions 484 and 417 in addition to 501, lining across this receptor binding domain of the spike protein, which is a key epitope for targeting of antibodies. So we have these different classes of antibodies that recognize the spike protein, and both class 1 and class 2 antibodies are affected by the mutations in these variants. Of course, there's another part of the spike protein called the N-terminal domain, which has also been identified as an antigenic supersite. In other words, lots of antibodies see it. And these Brazilian and in particular, the B1351 variant also have major deletions in that area, which we believe affect the ability of antibodies to see them. So there are many, many studies now, including some coming out from G2P Consortium and many from around the world, illustrating that these combinations of mutations which are present in the variants of concern, B1351 and the Brazilian variants, lead to escape from vaccine-induced antibodies. This particular one I rather like because of the color coding. You can see these different variants arising, and you can see here that the South African variants down here in red are the most affected by their mutations. They're most able to escape polyclonal sera raised after vaccination. And unfortunately, this appears to be translating into decreased vaccine effectiveness in the real world, where, for example, in South Africa, where clinical trials have taken place in the face of predominant circulation of B1351 virus, we see decreased effectiveness, both for the AstraZeneca vaccine against mild, which was almost showed no efficacy against mild or moderate COVID in South Africa. Novavax showed a decrease of around 30% in vaccine efficacy, and the Janssen also decreased down from 85% in other parts of the world to 54% in South Africa. So where we stand at the moment is, will there be a need to update vaccine against these, to take into account this antigenic change that we're seeing in these variants as we go forwards? If not now, then when and how? Just finish off really with some nice work again from a preprint, I think last week, trying to pull together a lot of different data from different studies to sort of get towards what we would love to have, which is a correlate of protection, an amount of antibody neutralization which made us confident would confer protection against different variants. And so what they've done here is that they pooled data from lots of different studies, normalized on convalescent sera, assuming that that's pretty much the same around the world, and then plotted this protective efficacy correlate. And then they've done some modeling to suggest how you can then use that to work out what the drop in neutralization would need to be to sort of affect the efficacy of your antibody or your vaccine against different variants. And I think this combination of antigenic cartography, which we're very familiar with in the influenza world, is something that's going to be very important going forwards to try to predict when we may need to update our vaccines. So I'll summarise by saying we can see a lot of variants, and COG-UK are revealing those and playing this wonderful pivotal role, really leading light in identifying the genomic changes. And what G2P aim to do is to try and interpret that information and to understand, using natural isolates, recombinant viruses and expressed viral proteins, what those genomic changes mean for our ability to mitigate against this virus. And I'd like to thank very much all the members of G2P, my partners in crime, Brian Charleston, Mike Malin and Massimo, and all the different institutions around the UK that have come together to try and pool our efforts. And we very much hope to learn from COG-UK's successes. So thank you very much for having me today.
Thank you, Wendy. That was absolutely fascinating. We are a little over time. I'm just going to do one very quick question, merge two together. Obviously, lots of interest around this, but do you think a universal SARS-CoV-2 vaccine would be possible, or are there any that are affected that you're aware of against the South African variant currently?
So I think it's important to say the vaccines are effective against the South African variant, as you saw from some of those data, just not as effective as we would like. And I think we've all been quite delighted by how well the vaccines worked when those clinical trials were revealed. And if we'd have started out a year ago with only 50 or 60% efficacy, I think we'd have been feeling very good. So I don't want to cause too much alarm. The vaccines do work, and if you give the right regime and the double doses, I think they will work. Nonetheless, I think that probably, some manufacturers already are thinking about updating to the South African strain in order to match with that, because that one seems to be the most antigenically distant at the moment. And I think going forwards, we're going to just need to keep monitoring that and working out how these different viruses can cross-react. In terms of a universal vaccine, I mean, I'm a veteran of the flu field, and that's been an aspiration.
Your holy grail.
It's a holy grail, and it's not easy. So I'd love it to be easy, but I don't think it will be.
Yeah, thank you, Wendy. Thank you very much. So we'll move swiftly on. Hopefully Nick is now with us. Yeah. So Dr. Nick Davies is going to talk more about the B117.
Yes, thanks very much. Sorry about that. My broadband failed right at 3 o'clock, which is
Are you able to do it? Oh, brilliant. Thank you.
Okay. Right. Okay, thanks very much, Wendy, for stepping in at short notice to reshuffle the order there, and apologies again. So I'm going to speak, as has been sort of ably set up by Judy, about B117 and severity. So with the identification of the B117 variant, this question that's sort of been percolating really throughout the COVID-19 pandemic has sort of arisen again, especially around sort of last December when it was first identified. And that's, given that the coronavirus is evolving, should we expect it to become more severe or less severe? So I've just gathered a few headlines here. And there have basically been sort of two perspectives on this issue for the most part. One being that we just don't know, it could go either way. And the other being that there is this sort of inexorable law of nature that states that inevitably as a virus mutates, it will become more and more benign to the host, which is sometimes called the law of declining virulence after some work done in the late 19th century. So I'm going to address these two perspectives using some quick evolutionary theory. So this is a model of pathogen transmission that was analyzed by Anderson and May back in the early 80s. I'm showing it here both in sort of flow diagram form and for the real diehard fans, the differential equations down here in the bottom left. But this is basically a simple model of pathogen transmission that has and tracks hosts in 3 different states: SIR model, susceptible, infectious, and recovered. Susceptible hosts become infectious upon contact with another infectious host at a certain transmission rate, beta, so that controls how quickly the pathogen spreads through a population. And then from the infectious stage, a host can either recover from infectiousness at this rate gamma, or die from the infection. So this is the mortality rate attributable to the infection itself at this rate mu. And so that's the system that I'm going to talk about in the next minute or two. And you can solve for the basic reproduction number of this pathogen in this system, and it gets you the answer R0 equals beta, the transmission rate, over the mortality rate attributable to the pathogen plus the recovery rate associated with the pathogen. So when we're thinking about variant fitness, mutant fitness, something which is approximately true is that if a mutant arises with an R0 which is higher than that of wild type, then selection will favor that mutant, and that mutant will eventually displace the wild type. So we can use that kind of framework to think about how a mutant that arises with changes to these parameters, the transmission rate, the mortality rate, or the recovery rate, might fare in competition with the wild type. So again, holding this equation for R0 in our heads, we can do something like posit that, okay, given that we are holding constant, say the recovery rate and the mortality rate, how does the mutant's fitness change with the transmission rate? Well, unsurprisingly, this kind of shows that as the transmission rate of a mutant increases relative to the wild type, its R0 increases, and so that mutant would be expected to do better. Similarly, we can hold constant the transmission rate and the recovery rate and think about how changes in the mortality rate will affect the R0 of a mutant. So here we see that as the mortality rate decreases, the R0 increases. And you don't really need an equation to tell you that. Essentially, the insight is that a disease which kills its host before it has a chance to transmit onto other hosts is going to be less successful in a competitive framework. So that's kind of that underlies this view, as Anderson and May themselves point out in this article, that all things being equal, assuming that a mutant could independently adjust these three parameters, a mutant with a lower severity would outcompete the wild type. So they said, were these parameters, and they're talking about these three in the equation here, not inextricably linked, the ESS, which is an evolutionarily stable strategy, would clearly favor mortality going to zero. They go on. Insofar as there is a basis for the common view that successful parasites and pathogens are harmless, this is it. But the biologically based interlinkage among the transmission rate, the mortality rate, and the clearance rate invalidates this simple argument. So what they mean there is that it may not be very realistic to assume that these parameters, these different phenotypes of a given virus are independently adjustable. So let's return to our equation here for R0 and suppose that rather than the transmission rate and the mortality rate being something that the virus can adjust independently, let's replace the beta and mu here with functions of some other underlying phenotype, which we'll call V for virulence. Okay, and let's, for sake of illustration, assume that the mortality rate and the transmission rate of a given mutant depends upon this underlying characteristic of its virulence. So we might have, for example, for the mortality rate, a sort of linearly increasing function of virulence. And for the transmission rate, say I've drawn here as an illustration, an increasing function of virulence, but which saturates. And there's a lot of different reasons why transmission rate might saturate with virulence. For example, a person only makes a certain number of contacts per day. So there's an upper limit to how quickly, or to the number of people that they can spread an infectious disease to. And so if you make these two parameters reliant upon some underlying property of the mutant, you get this kind of inverse U shape for R0. So you find that the maximum fitness for a mutant which has emerged under these assumptions is under an intermediate level of virulence. So it says, yes, if, under these assumptions, if an infectious disease arises with a very high virulence, we might expect natural selection to favor that amount of virulence to decrease because the R0 will be higher with a lower virulence. But if a virus emerges with a low virulence, we could equally expect that to increase to this sort of intermediate level. So that's sort of just underlying the basic idea of why this picture that mortality will necessarily evolve to be lower over time is potentially a bit simplistic if both mortality and the transmission rate are controlled by the same sort of physiological, if you will, properties of a virus. So that's the theory aside. I'm now going to talk about sort of how we address this question. Does B117 cause more severe disease? So the data that we had to analyze were 1 million positive community tests in England with and without S gene target failure, which has been mentioned already as a proxy for B117. You can see here that it's illustrated in England for lower tier local authorities, the spread of S gene target failure over time. This was linked to a line list maintained by Public Health England of COVID-19 deaths in England. And we also had the age, sex, ethnicity, whether the individual lived in a care home, their index of multiple deprivation, which is a measurement of socioeconomic deprivation, their lower tier local authority of residence, and the date upon which they were tested. So these are the data that we had. You can see here the number of samples over time, clear spread of S gene target failure as we get into December, January, and February, and the number of deaths associated with tests on a certain specimen date over time. So when we just looked at the crude survival in these two groups back in January, we found that there is sort of little difference between them. And if anything, the survival was actually slightly better in the S gene target failure group. So that motivated us to look a bit more carefully using a statistical model at what might be going on here. So we used a Cox proportional hazards model, which is used to estimate the relative hazard, which means the sort of relative rate of a given event. And that event is measured in different strata of a population as a function of some covariates. So the particular stratification we used was looking at only individuals that were tested who lived in the same lower tier local authority and were tested on the same day. So comparing the relative mortality between individuals that live in the same place and were tested on the same day. And that's important because we did that in order to account for changes in testing rates that might happen over time that might be correlated with the spread of B117, and also changes in hospital pressure over time that again might be correlated with B117. And as covariates, we added into the model not just the covariate of interest, their SGTF status, but also the age, sex, ethnicity, care home residence, and index of multiple deprivation. So when we did this, we isolated an increased relative hazard of death, as Judy mentioned, is sort of 55% higher among individuals that were tested in the community. And we found no significant difference across any of those covariates. So no evidence that the hazard of death with SGTF was any different for individuals of different age groups or sex or ethnicity and so on. And although we initially had this picture where it was kind of unclear from the data themselves whether there was a difference in mortality, as we accumulated more data and looking back from February, we did eventually see that this pattern, apparent pattern in the crude survival, reversed. The explanation for that is that SGTF was initially spreading more among young people, so that the SGTF group was biased to be slightly younger. And of course, mortality in younger individuals is substantially lower. So those are our main results. We did lots of sensitivity analyses, which I'm not going to go into, and we presented our results according to absolute risks as well as the relative risks. So for example, illustrating that for an individual test in the community who's, say, female and 85 or older, their absolute risk of death would go from 13% to 19 or 20%. So this is my last slide. So that's sort of our one study, but as Judy indicated, there have been a lot of different studies looking at this question, and I've sort of gathered them together here in this table. And yeah, as Judy said, the results really do look differently depending on whether you look in hospitalized patients or in individuals in the community. So maybe the best unbiased estimate of the severity of B117 you could get would be by looking at a random sample of people who are infected with one strain or the other and comparing their probability of being admitted to hospital or of dying. So the ONS has apparently started this, and they reported that they got a hazard ratio above 1, but they didn't give any kind of point estimate for that. So that's kind of the, I think if this study does get finished, we'll get a really definite answer to this question. There's a number of studies now that have looked at the probability of dying or of being admitted to hospital or of being admitted to ICU given a positive test in the community. And generally, these have all found very similar results, that the hazard ratio is above 1, meaning there's an increase in those in the likelihood of those events happening given a positive test in the community. There's a few maybe slightly outlying studies which don't identify a huge increase or which aren't significantly differentiable from 0. But then the hospital studies, which is all the ones over here, kind of identify odds ratios or hazard ratios that are not statistically distinguishable from 1. So it's a bit of a difficult and confusing question. I mean, one way you can explain this pattern generally is by hypothesizing that B117 makes you more likely to get admitted to hospital, but then once you're in hospital, there's no difference in the outcomes in terms of either death or ICU admission, or even death in the ICU. So that's one way of looking at all these studies and drawing a conclusion that accords with all of them. But I think the jury is still out, essentially, and that's the answer to the question, true or false. It's I think that we need more evidence to really be sure of it. I won't talk about biases in testing. So thanks very much for listening. These are the two papers that we've put out looking at the different properties of B117. And I'm sure I've run out of time, but happy to take questions. Thanks very much.
Thank you very much, Nick. Another fascinating talk. We'll just have one very quick question before I hand back to Sharon for a final roundup. How do you account for how treatment has changed, and could this be a factor that's masking any increase in severity?
Well, because we only compare the probability of dying between individuals who lived in the same lower tier local authority and were tested on the same day, we kind of take that as a proxy for those individuals receiving the same standard of care, just that they're likely to be admitted to the same hospitals, and that you know, you don't know what your status, the people treating you don't know whether you have B117 or not, and so you would likely receive the same standard of care. So we don't actually compare between individuals at different times, just within the same sort of stratification of time.
Okay, thank you. There are more questions, as I say. I'm sorry that we can't ask all of them, but I'm going to hand back to Sharon now just to do a final roundup of what's been an absolutely fascinating afternoon. So I'd just like to say thank you to all the speakers for keeping to time and negotiating the IT issues.
Thank you very much. Well, what a great afternoon it's been. I've hugely enjoyed it. With Zoom, once you start to use it over a year or so, it's inevitable that there are some glitches, but I think everybody handled that brilliantly. And the most important thing was that the data that we saw today was absolutely fantastic and really top class. So thank you. So I'm just going to close by thanking colleagues, and I think that's such an important point before you all leave. First of all, I would like to thank very much Elaine Westwick and Andy Jeremy, who organised the meeting today, and they did a tremendous job steering us through everything. Thanks to the chairs, that's Catherine, David and Sally, and also to all of the speakers. A massive thank you to the consortium members of COG-UK. There are around 600 now, and it's you that make the difference to how we function and how much the data that we generate. So a massive thank you to you. I'd like to thank you all for attending today. There will be another showcase event in the near future, so please look out for that, and I hope that we'll see you back again in the very near future. So thank you and goodbye.